So apart from this non-cyclic electron transport system, one more is there, that is called as a cyclic electron transport. So this cyclic electron transport is a source of additional ATP which is required by the Calvin cycle. So in this process, okay, this was studied by Frankel in Rhodospirillum rubrum bacteria. In this cyclic electron transport system, only PS1 is operated, and here the electrons which have been lost from PS1 will finally reach the PS1. So that's why we call it as a cyclic electron transport system. In this one, the P700, that is the reaction center of photosystem 1, will get excited by absorbing light at 700 nanometers and losing the electrons. So it is shown as a P700 with a star mark, and here the electrons which have been lost from the excited form of P700, those electrons are moving through different electron carriers like ferredoxin. From there they will be moving to the cytochrome B6. From cytochrome B6 they will enter into the plastoquinone. Quinone cycle will be operated here. From there it will be moving to the cytochrome F. From cytochrome F, those electrons are moving to the plastocyanin. From plastocyanin, the electrons once again move to the reaction center of PS1, that is P700. In this process, the quinone cycle is operated. The quinone cycle is operated between cytochrome B6 and cytochrome F. So likewise here, the primary acceptor in this cyclic electron transport system is a ferredoxin, and finally those electrons are moving to the same reaction center, that is P700, from where the electrons have been lost. And in this process here quinone cycle is operated, where when one electron is moving through the cyclic electron transport system, two protons are translocated or moved into lumen from stroma. So here in this process, the protons which have been accumulated into the stroma, it also leads to the proton gradient. So proton concentration gradient is established. So this proton concentration gradient is responsible for pushing of or movement of the protons once again from lumen to the stroma side. So in that process the ATP will be formed, and here in this process photolysis of water does not take place. At the same time, no evolution of oxygen, because when photolysis of water will occur, then only the oxygen evolution will be taking place. But here, as a photolysis of the water does not take place, there is no evolution of oxygen, and no NADPH formation, and only ATP formation is there. So these are some of the differences that we can find between the cyclic electron transport system and the non-cyclic electron transport system. And here we had discussed about DCMU, which is inhibiting the transport of electrons between PS2 and PS1, but here as there is no role of DCMU, because only PS1 is operated in this process. So here this type of process where ATP formation is taking place because of a cyclic electron transport, we call the process of ATP formation here as a cyclic photophosphorylation process.